I'm a neurologist at the University of Pennsylvania, and as many of you know, I'm one of the people who's working with the CMTA on the projects involved with STAR. And what I have been tasked to do today is to give you an update on where we are at with the treatment for some of the type 2 CMTs. So I want to give you a couple of disclosures just because I think it's the right thing to do. I have been funded by the NIH and by the CMT Association to do work on both animal models of CMT and in clinical trials involving the natural history of different kinds of CMT. And I have served as a consultant for some of the companies that will actually be presenting on today who are trying to find treatments for CMT2A. So just to get us all on the same page, I want to start off by telling you that CMT is a very diverse group of diseases, at least 100 known genetic causes that can be inherited in different patterns, sometimes dominant, and those are usually called CMT2, but even recessive forms. And the commonest forms of CMT2 that we have decided to focus on at the CMT Association include CMT type 2A, CMT type 2E, and CMT type 2F, though there are other projects and other forms of CMT that I don't have time to discuss today. But if you want to ask me about them or anything that I present during the talk, at the end of this, I'll look in the chat and see if I can find your questions and address them if there's time. And if there's not time, I will get back to you after today's session is over and answer you individually. Okay? So the purpose of this slide is to introduce you to where the action lives when it comes to neuropathies. Whoops. The neurons that supply sensation to the body live in little clusters outside of the spinal cord. This is the spinal cord. And these are called ganglia. And the sensory neurons live in these ganglia. And the motor neurons, which supply the muscle and make the muscle work, actually live within the spinal cord in this region right here. And so I want to give you this pictorial graphic and emphasize that the neurons live in different places because the goal of different therapies under discussion today is to give something that the motor neurons need here in the spinal cord or that the sensory neurons need up here in the dorsal root ganglia. We have to deliver either by a virus or another route some molecules that will make these cells function better such that we can stop the progression of the neuropathy. So these are the neurons that we need to fix. The sensory neurons here and the motor neurons here. And this can be done actually quite effectively, as I'll show you later. The commonest forms of CMT are, for the type 2s, are those caused by mutations in a gene called Mitofusion II. And third or fourth on the list would be mutations in a gene called neurofilament light, which causes CMT type 2E. At the very end of the talk, I'm going to get to a CMT2F, which is caused by mutations in a gene called heat shock protein beta 1. We'll mention but not elaborate on the fact that we have one particularly common recessive form of CMT called CMT4A. It's caused by recessive mutations in GDAP1. And there's another form of CMT that you can hear about from Dr. Stefan Zuckner in a different session, which is caused by recessive mutations in a gene called SORD. So these are recessive mutations, and I'm not really going to emphasize much about them today, except that theoretically they are treatable in quite the same way that I'm going to talk about for the commoner types of CMT type 2 listed above. So the first thing I need to explain is what we think Mitofusion II does. This is the gene that's mutated in CMT type 2A. And what we know about the function of this gene in the main is that it sits on the outer surface of an organelle in our bodies called mitochondria. And mitochondria are basically the energy creators for all cells in our body, and neurons are no exception. And so this gene encodes a protein that sits on the outer membrane of the mitochondria, and the two opposed mitochondrial membranes use this interaction to allow them to fuse. And we think that this is quite important for the function of mitochondria, such that if this Mitofusion II gene isn't working right, the mitochondria fail to fuse normally, and ultimately that leads to their demise. And without mitochondria, no cell can live, and certainly not neurons. And so the lack of proper Mitofusion II function, I think, ultimately causes the failure of mitochondria, and that in turn results in energy failure, and that in turn results in axonal degeneration. So that's the premise that we're thinking is the means by which mitofusin normally works, and then when Mitofusion II has a mutation, then it doesn't work, and the axons are what suffers from that problem. If I can go back a slide, the axons are these structures that are hooked from the neurons, the motor neuron here and the sensory neuron here, and they are the living extension of this cell in the spinal cord or dorsal ganglia that goes long distances in our body, up to 2 or 3 feet, to connect motor neurons to muscle and to connect sensory neurons to sensory appendages, say in the skin or elsewhere in the body. So if these axons lack energy and they die at some point because they lack energy, then loss of sensation and loss of movement will be the consequence of that. So I should tell you that there are many, many different Mitofusion II mutations that have been described. They all the ones on this slide and many more, result in a clinical picture of neuropathy. And sometimes it's a really severe neuropathy that begins in childhood. Other times it's a milder neuropathy that begins even in middle-aged adults. So it's not every mutation has the same consequence, but they all ultimately cause neuropathy of some severity. And some of them, believe it or not, in addition to affecting the axons in our nerves, affect the axons in our eyes and result in a phenomenon called optic atrophy, which means your vision is impaired. And you can see that these mutations are scattered all along the length of the prediction of the protein, or the predicted structure of the protein. So disrupting the function of the protein in many, many places results in compromised function. I want to mention 2 of these mutations in particular, because these 2 mutations are the ones that were selected by the CMT Association to be made into a rat model. And here they are. It's histidine 361 tyrosine and arginine 364 tryptophan. And these 2 mutations individually cause a severe neuropathy that begins in childhood. And we picked them deliberately because we wanted to make an animal model that would give us the best chance of seeing a neuropathy. So we've taken several approaches to treating CMT2A, working with our partners in industry and in academia. And the one that we'll talk about first is the idea that we're going to try to fix the broken gene. And so this is work that's been done in a lab led by Jim Wilson, who's at the University of Pennsylvania, and through a company that's been founded by Dr. Wilson and his colleagues called Passage Bio. They've been working on ways to basically replace the mutation that's causing the neuropathy in CMT2A with a normal copy of the Mitofusion gene. And if that were to work, then that would theoretically likely rescue the function of the Mitofusion II, because now you'd have a normal copy and not a mutant copy, and that would provide a fix. A second strategy, which is in the news very much these days, is to use a different strategy, which is to use a CRISPR-Cas9 strategy. And this is work being led by Drs. Conklin and Judge at the University of California in San Francisco, which is actually where CRISPR-Cas9 technology was invented and pioneered. And they're using this technology, which I can't really explain to you on this slide. You basically directly try to either delete the bad copy of the Mitofusion gene, which is, I think, the strategy that Dr. Conklin and Judge are taking, or more audaciously, it's conceptually possible that you could even try to fix the bad copy of the Mitofusion II gene to repair it entirely such that it work properly. That's a harder to do proposition, but not impossible. And those are technologies that are available through CRISPR-Cas9. And it's different in its design, but not different in its intent than fixing the mitofusin gene in the way that Doctors Wilson and Hinderer intend to do. Another strategy, completely different than what I've discussed, is to use small molecules to enhance the function of the Mitofusion II mutants. And this is work that's being led by Dr. Gerald Dorn at Washington University in St. Louis through a company that he founded called Mitochondria in Motion. And there, the idea is that small molecules can enhance the function of the mutant Mitofusin II protein and make it work better, and thus sort of bypass the genetically damaged Mitofusin II protein. A third, and again, completely different strategy, is to use a drug that would inhibit a molecule called SARM1. And there are several companies trying to develop such drugs. And the strategy of inhibiting SARM1 would be to prevent axonal degeneration. And this would be a strategy that would not only work for treating CMT type 2A, but maybe many other kinds of CMT2, because axonal degeneration is a final common pathway of many, many neuropathies, including many inherited neuropathies. And on my last slide, I'll at least mention there are other small molecule pharma companies that have partnered with the CMT Association to use the CMT2A rat model for unique strategies that these companies have come up with, and I don't really have the time and even the necessary knowledge to tell you about them today. And in the background and in parallel to all of these efforts that we're currently doing would be the idea that we're trying to develop knowledge around how CMT2A progresses in people with CMT2A, such that if one of these therapies were to become available for a clinical trial, we would know how to measure progression in CMT2A in the clinical trial for the purposes of showing whether the treatment worked in people. And that's going on in parallel at this time in a grant that's been funded by the CMT Association. So I want to mention this wonderful rat model that the CMT2A developed. They've actually made two, and I'm only going to show you one of them here. And basically, it develops a neuropathy, which is amazing because not all animal models do develop neuropathy. And the way I'm going to give you an idea of how this works is you can measure the nerve conductions in the tail of a rat. This is a schematic tail of a rat, and you can, just like some of you have had EMGs of your arms, you can do an EMG of the tail. And normally the size of the sensory response gets bigger and bigger as the rats age from 8 weeks to a year. But in the CMT2A mutant rats, it increases for a time, but then actually decreases. And what you see if you look at the tail by histology, if you do, microscopy of the tail nerves, which is what I'm showing you here in the normal littermate and here in the mutant littermate, you can see that there's been a loss of these myelinated axons in the CMT2A mutant rat. So this is a big deal. So that means by both serial nerve conductions and by sacrificing the animals and looking at their tails, we can prove without a doubt that a therapeutic intervention either prevented axonal loss or ameliorated it, or it didn't help at all. And having an animal model that accurately depicts the efficacy of a therapy is a godsend for going forward in clinical trials, because you would want it to work in a rat that has CMT2A before you would try it in a person with CMT2A. So how we think this would work in a rat model and in a person with CMT2A is that we would inject the virus or the therapy that was virally mediated into the spinal fluid that would then have access to both the motor and the sensory neurons that I've already shown you. We would do it in a rat model before the onset of the neuropathy, say prior to 8 weeks, and then we would let the animals age, and if the therapy worked, instead of having the nerve becoming degenerated like the one in the lower panel, the nerve would remain healthy, and the nerve responses would remain healthy as well. And so if we were able to see those responses, we would be very encouraged to think that the therapy had worked. In the case of the mitochondria in motion molecules that are intended to enhance the activity of Mitofusion II, then the animals would be dosed either orally or by some means of injection, but not necessarily into the spinal fluid, with a molecule that's designed to enhance the function of Mitofusion II. And again, I think the best initial trial would be to do it before the onset of the neuropathy and see if, in fact, we can prevent the nerve from degenerating like this one and remaining healthy like here. So having an animal model, a rat model of CMT2A enables these kinds of things to be studied. It's a lifeline to the scientific progress for treating CMT2A patients. So now I want to switch gears. I want to talk about CMT2E and then talk about what our strategies are for CMT2E. Well, the first thing to tell you is that dominant mutations in a gene called neurofilament light cause CMT2E. And the normal function of neurofilament light is to encode a structural protein found in axons called neurofilaments. And I've circled a bunch of them here in this electron micrograph. These little dots are the neurofilaments. And if you look at an animal model that lacks neurofilaments, an animal model of CMT2E, therefore, you don't see those little dark spots. You see the microtubules, which are a bit bigger, but you don't see the neurofilaments. So to a first approximation, it looks like CMT2E is the consequence of having no neurofilaments, and the consequence of having no neurofilaments is what the neurofilament light mutations typically do. So that's what we want to fix. So I should tell you, just like for Mitofusion, there are many different mutations that have been described of neurofilament light. Some of them are particularly bad, and so they're shown in red. And the one that we've modeled in the mouse is one of these bad mutations, which causes a childhood onset bad neuropathy. So that's a mouse model that's been made. And I'll go a little faster over this because we talked about it for CMT2A. We have the same ideas about we can try to knock out the bad gene and replace it with the normal neurofilament light gene. And that's work that's being proposed by Drs. Brown and his colleagues at Ohio State University. We could try to use the CRISPR-Cas9 system to delete the bad copy of the neurofilament light gene. And that's work that Dr. Conklin and Judge are doing at UCSF. And again, theoretically, you could even intend to actually fix the bad copy of neurofilament light. But again, that's a bit farther down the road and much harder to do in any case. We benefit from this animal model that I briefly mentioned of neurofilament light mutations causing CMT2E, and I'll show you a slide in a second. And for neurofilament light, we have some additional help in getting an answer or a treatment in that we've gotten human stem cells to be developed from fibroblasts, and these can be used to screen drugs that might help neurofilaments form. And this is a project that was done by Dr. Ronald Leem and also by Dr. Mario Saporta at the University of Miami. And Dr. Saporta has actually shared his stem cells with the Conklin lab to allow them to initiate their studies with CRISPR. So the CMT2A has supported all of this work, and we obviously are collaborating in and amongst ourselves and with any other scientists in the world, for that matter, to get to these goals. This is very similar to what I showed you for the mutations of Mitofusion II that cause CMT2A in the rat. This is a mouse that has CMT2E. It's a particularly bad mutation, as I mentioned a moment ago. And if you look at the tail responses of the normal mouse, they get bigger as they age. But if you look at the tail response of the CMT2E mouse, you can see that they get smaller as the animals age. And if you look at the nerves as the animals are 48 weeks old, you can see that there are many, many fewer myelinated axons in the mutant animal than in the wild type. So any treatment that you would give, you'd probably give early, say here, before the onset of disease, and hope that you could see that you prevented the nerve from ending up like this and keeping the nerve more normal. So again, having an animal model of CMT2E is a very important and almost essential piece to linking therapeutic events or therapeutic ideas that you can do in animals and before you would actually approach doing that in a human. So I want to mention a couple more things to give you a different take on all of this. So I've already mentioned the work at UCSF having to do with CRISPR-Cas9, and I mentioned that there were stem cells that have been used by this group. And so the way that that analysis could work is that you could take the patient's stem cells and you could actually convert them into neurons, and you could test the effects of the treated stem cells and treated neurons as to whether they developed sort of the equivalent pathologies that you see in CMT2E mice or patients. And if you were successful in knocking out the bad copy of the gene, or even more audaciously fixing the mutant copy, then you could see an improvement in the morphologies of the neurons that you culture from the stem cells, and similarly improvement, as I described in the last slide, in the nerve structure of the animals that were treated with the CRISPR. So this is something that you've heard about in the news many, many times. It's possible to consider doing this for different neurologic diseases, including CMT, and we're very anxious to see what these results look like, and we'll share them with you as we get results from the group at UCSF. So I do want to mention that the CMT2A has now successfully made a rat model of CMT4A, which are the recessive GDAP1 mutations. And we are hoping that they develop neuropathy, but we don't yet know whether they do. I want to tell you that there's been a lot of work historically on a protein called HDAC6, which I don't really want to spend too much time talking about, but inhibiting HDAC6 has been raised as a possible way of treating different kinds of CMT, and this is work that's being investigated by Dr. Robert Burgess at the Jackson Laboratories. And we're also using Rob Burgess's expertise in the Jackson labs to test whether SARM1 inhibition can work, and that's actually being tested on a variety of different kinds of CMT by Dr. Burgess. There are many partners that are looking at our animal models of CMT2A and CMT2E in particular, with therapies that are privy to their discovery. And we are allowing them, through a clever use of licensing, to test whether those drugs work in these animal models by licensing the animals or developing them ourselves and having them tested either by the investigators themselves or through a contract research association. So just to say in a simple way that we are enabling anybody, any company, any investigator to use our animal models for the purposes of finding treatments for CMT. And I think this is the last slide, and it just emphasizes the parallel effort that's being made to find in people attributes that we can look at and measure and quantify that would tell us whether a drug or a therapy was working for different kinds of CMT. So one concept that I want to leave you with is you'd like to be able to show that your therapies engage the target that you intended to. So for some treatments, this may be possible. I will mention work by John Severin and Mike Shai in CMT type 1A, where that's caused by the overexpression of PNP22. And John and Mike have been able to show with skin biopsies that you can measure the overexpression of PNP22 in people with CMT1A. So a treatment that worked effectively for CMT1A would, in fact, be able to be measured in the skin biopsy. Sorry about that. Secondly, you can do electrophysiology of people and skin biopsies of people with different kinds of CMT. And if that was going to show you that you had prevented axonal loss, you could detect that directly by doing nerve conduction studies in people or doing skin biopsies of people, say, before and after treatment. More difficult, but still on the list of things that can be done, is to show that the neuropathy is improving. So that could be done theoretically by a clinical exam, the CMT neuropathy score. It could be done by measuring the fat content of muscle that's being affected by the CMT. And as the CMT gets worse, the fat content goes up. And Mary Riley and Mike Shai have shown that fat content increases in CMT. And so once we have a treatment, I think the goal would be to show that the fat content doesn't increase. And that would be an indication that things are holding their own and the treatment is working. And lastly, David Herman in the CMT Association support is looking at patient-reported outcomes, like what can people sense about themselves that might inform us as to whether the therapy is working. And so that's another very important and theoretically possible way of doing it. So you have to do these clinical assessments in parallel, because how bad we would all feel if in fact somebody came up with a reasonable therapy that we thought was working in our animal model, moreover, and we weren't ready to do the clinical trial in people. So having these efforts being made in parallel is absolutely important to making progress as fast as we can and as fast as you want us to do it. So I think that that's all of my slides. I have a couple minutes that I didn't go over time with, and if there are any questions, I can try to look in the chat box, but that would require me to be more technically gifted than I probably am. So maybe somebody on the tech side could fix my slides and show me what the chat box looks like.
Yes, we can definitely help out with that. There are a few questions that we've had come in. We've got about 3 minutes, so
okay.
Let's see here.
So I can see it now. So the first question I see is from, I have CMT1B. Anything in the pipeline for us? Thank you, Steve. So I would tell you that I'm not talking about CMT1B in my talk, but there is a program funded by the CMT Association that Maurizio D'Antonio is doing in Milan. And Maurizio has identified how to manipulate a thing called the unfolded protein response, which is usually activated in CMT1B. And so it is possible that we can manipulate the unfolded protein response to make CMT1B better, in other words.
Okay.
I don't think I can scroll down on this, so maybe that's it.
Yeah, it looks like there's a couple. I'll put a couple on stage. One is 2F was listed but not discussed. Also, what is a vision impairment? Is impairment seen in 2F?
So the CMT alphabet's confusing even for me. I believe CMT2F are the dominant mutations of heat shock protein beta 1. That's a reasonably common form of CMT. It's mostly a motor problem that sensory function is largely not affected, but motor function is definitely affected. And for heat shock protein beta 1, I think the biggest thing to be tested there is whether the HDAC6 inhibitors work. There is a paper showing that in a mouse model, HDAC inhibitors make CMT2F better. And I'm not really able to say much more about that without sort of rereading all the stuff that's known, but that's, I think, the main breakthrough with 2F that's come to light to date. There may be an effort underway to look at what we call the natural history of people with CMT2F, which is to say, how do they progress over time, using that sort of analysis that I mentioned in my last slide.